Okay, so descent of the testes. Okay, this is very important. In the male fetus, during embryogenesis, the testes actually originate from masses of tissue behind the parietal peritoneum near the kidney. At about 2 months before birth, as a result of testosterone, okay, it will trigger the developing testes to basically descend into the scrotum. Okay, we have the gubernaculum. The gubernaculum. This is going to help in the descension process through the inguinal canal, which is this is right here, the inguinal canal. After this, the spermatic cord. The spermatic cord. It's literally like a cord, and inside it, it contains the ductus deferens, which is the vas deferens, and blood vessels and nerves are connected to the testes. Now, failure to descend the testes results in cryptorchidism. Cryptorchidism. This is a congenital abnormality where the testes are not descended. Okay, and it's usually fixed by surgery. If it's not fixed, this can lead to infertility. So it needs to be fixed within the first year. Structure of the testes. So the testes has a fibrous capsule. Each testis is enclosed by a fibrous capsule called the tunica albuginea. From the tunica albuginea, there are septa that go into the testes. So think of it like this, and then these septations go inside like that. So these are septa, and they make compartments called lobules. So each one of these is a lobule. So this is a lobule. This is another one. This is another one. Inside each lobule are these highly coiled tubes called seminiferous tubules. These are highly coiled tubes. These seminiferous tubes are lined with a special type of stratified epithelium, which contains spermatogenic cells. These spermatogenic cells actually give rise to sperm cells eventually. Okay. Now, in the areas between the seminiferous tubes, we find interstitial cells, also known as cells of Leydig. These guys produce and secrete the male sex hormones, like the testosterone. So here you could see a good picture. Here you could see the tunica albuginea, and then you can see these are the septa, and each one of these is a lobule. And inside the lobule, you can see the coiled tubes, which are the seminiferous tubes. Okay. Then if you take a cross section or a plane section of the seminiferous tubes, this is the lumen of the seminiferous tube. This is the basement membrane of the seminiferous tube. And here you could see the sperm cells. In between the seminiferous tubes, you would find the interstitial cells. Okay, so formation of sperm cells. Formation of sperm cells. Okay. Sustenticular Sertoli cells. These are large cells that are found throughout the entire thickness of the epithelium of a seminiferous tube. The sustenticular Sertoli cells, their function is to support and nourish spermatogenic cells throughout their development into a sperm. Without Sertoli cells, spermatogenic cells cannot develop and become a sperm. So sperm formation occurs in the following sequence. It starts out with a spermatogonia, which becomes a primary spermatocyte, which becomes the secondary spermatocyte. Then that develops into spermatids, which finally becomes spermatozoa. Remember, sperm cells only have haploid or half because they are produced through the cell division process of meiosis. If you are ever asked to identify or define what meiosis is, it basically means cell division that reduces the number of chromosomes by half. The next two terms are important. Spermiogenesis. This is the process of developing spermatids into sperm. Spermatogenesis. This is referring to the combined process of both meiosis plus spermiogenesis. Okay. So spermatocytes, they will come from spermatogonia. Spermatocytes, they will come from spermatogonia. During spermatogenesis, we will produce 4 haploid sperm cells. So let's go over this. Here we see the primary spermatocyte. Remember, this came from a spermatogonia. Primary spermatocyte. Primary spermatocyte, remember, this is haploid. Okay. Primary spermatocyte undergoes meiosis 1. And at the end of meiosis 1, it becomes 2 secondary spermatocytes. Remember that the secondary spermatocytes are haploid, 1N. Each of the secondary spermatocytes undergoes meiosis 2. At the end of meiosis 2, we get 4 spermatids, also haploid. And then each of the 4 haploid spermatids mature to form and become a mature sperm cell. Make sure you know the steps and make sure you know which ones are haploid and which ones are diploid. Again, this is the process. Okay, so during spermatogenesis, the spermatogonia cells, will replenish themselves and give rise to the spermatocytes. which develop eventually to a sperm.